Let me start recording. Anyways, this is the slide I believe we were talking about last time. So DNA is a polymer of nucleotides, and there are 4 different nucleotides in DNA. You should know them as A or adenine, T or thymine, C or cytosine, or G or guanine. I think we've talked a little bit about the flow of information. DNA stores information, and this information flows within a cell. This is actually the central dogma of biology, that the information is encoded in the DNA, and that information flows to RNA in transcription, and then flows to protein in translation. Each of the information at each step is coded in its own language, and we'll talk a little bit more about that, meaning the DNA is encoded in a language in DNA and RNA and in protein. You can talk about the flow of genetic information within a cell, between generations of cells, and for humans, that would be between generations, like from the information flows from the parents to the offspring. And you can also talk about the information flowing between cells of the same generation. Within a cell, the information flows from DNA to RNA to protein. Between generations of cells, the information flows from the parent cell to the two daughter cells. And that happens by the DNA replicating itself. So one chromosome then replicates itself, and then it's 2 chromosomes. And then the 2 chromosomes move towards opposite ends of the cell. And then the cell splits down the middle to become 2 daughter cells. And that's the way that most bacteria replicate. We'll talk a little bit more about that. It's also the way our cells go through mitosis. It's not the way our cells go through meiosis, and that's a little more complicated with humans. You have 2 parents, and they give rise to 1 offspring, or usually 1 offspring. Any questions about the flow of information between generations of cells? If not, let's talk about the flow of information between cells of the same generation. The information leaves a donor cell. Usually, but not always, what happens is the DNA leaves this cell after the cell usually dies. It doesn't have to be, but usually the cell dies, and then the DNA is naked DNA outside the cell. It is taken up by a recombinant cell, and then that DNA is expressed in the recombinant cell. And we'll talk about later in this lesson the 3 ways that DNA can leave one cell and then enter another cell. But that's the flow of information between cells of the same generation. Any questions about any of that? You should note that I've altered the figure from the book a little bit. I crossed this out and I added that in there. And that's because it's a little more complicated. The DNA leaves this cell and then enters the recombinant cell. All right. Let's talk a little bit more about the flow of information within a cell. This is called the vertical flow of information. The horizontal flow of information is this one here, where DNA flows from the parent cell to the daughter cells. Anyways, the language of the information is stored in DNA is in the nucleotides of the DNA. And there are 4 alphabets in this language, meaning there's 4 nucleotides that encode the information in DNA. In RNA, the information is encoded also in nucleotides, but these nucleotides are a little different. They're RNA nucleotides. These are DNA nucleotides. Once again, there's 4 letters in the alphabet, meaning the information is encoded only in 4 RNA nucleotides in RNA. In protein, the information is encoded in the amino acids, and there's 20 different amino acids or letters in the alphabet of protein. For comparison, English has 26 letters in its alphabet. Any questions about any of this? So in the DNA, we could say the information is stored as, that is a dog, the original information in DNA. After transcription, the information would be stored in a similar way, but with a different accent. And it would be something like, that is a dog. And then in protein, the information would be stored in a different language, and it would be quello è un cane, the same information, just in a different language. Any question about any of that? All right, if not, let's talk about the vertical flow of information, where DNA flows from a parent cell to 2 daughter cells. Or if we're talking about humans, DNA flows from the parents to 1 offspring. Any questions about any of that? All right, DNA flows from the parent cell to the daughter cell by the DNA replicating. And once again, the DNA copies itself, and then 1 copy of the DNA moves to one edge of the cell, the other moves to the other edge of the cell, and then the cell divides down the middle so that each daughter cell gets a copy of the parent's DNA. And we're talking about cells here. Now, obviously, with humans, it's a lot more complicated, and the mother and the father only give half of their DNA to the offspring. And we're not going to show that, but that happens in meiosis, where the eggs and the sperm have half of the parent's DNA, which does replicate similar to this. It's just that the eggs and the sperm only get half the DNA. And then the egg and the sperm unite to form the fertilized egg, which will become the offspring. How that happens is DNA replicates. Let me blow this slide up. By the double strand of DNA separating and become single-stranded, and then each single strand is used as a template to build the new strand of DNA. And so this is a template for this new strand, and this is a template for this new strand. We call the DNA replicating this way semi-conservative, where 1 strand is the original strand, which is used as a template to make the newly synthesized DNA. And how that happens is the polymerase binds to, come on, mouse, this nucleotide sees it's a T, and so it installs the complementary base pair, which is an A, on the new strand. And then the DNA polymerase moves down the strand, sees a G here, and then adds a C there. And it just keeps moving down the template strand, adding the correct complementary base of DNA. We call this DNA replication semi-conservative, where if the original parent DNA would be 2 black strands, then the daughter cells each get 1 original parent strand, the black strand, and then have 1 newly replicated strand of DNA, which is the white strand. Semi-conservative. This is opposed to the conservative model, where 1 daughter cell would get 2 black strands, and the other daughter cell would have 2 white strands. The conservative model of DNA replication does not happen. DNA replication happens by the semi-conservative model. And don't ask me what the liberal model would be, because I don't know. Any questions about any of this? All right, so I mentioned before that the DNA polymerase binds to the template strand here and sees an A there, and then adds a T to the new strand. How that happens is this will be a nucleoside triphosphate that's added, so this would be thymine triphosphate. And then this bond here breaks, and that will release energy because the phosphate, in this case, a diphosphate bond right here, has high energy in it. And when that energy is given off, that allows the reaction of adding this T here and then binding the phosphate there to that OH group there, meaning that the T, when it binds here, it will actually form a covalent chemical bond to the single strand that it's binding to, meaning this phosphate will form a covalent bond to this OH group here on the sugar group. Come on, mouse. My mouse died. Just a minute. There it is. And so the single strand of DNA will be held together by covalent chemical bonds. The double strand will only be held together by hydrogen bonds. And there will be hydrogen bonds between each base on a single strand, and it'll hydrogen bond to the complementary base on the template strand of DNA. Any question about any of that? And so here we go, binding that breaks off. The 2 diphosphates are here. Let's call it a diphosphate rather than pyrophosphate. And then the phosphate binds to this OH, and that makes the covalent bond for the single strand of DNA. And then the T will hydrogen bond to the A, and then this single strand will only be hydrogen bonded to this single strand, and there will be between the nucleotide bases, the hydrogen bonds. Any question about any of that? All right, if not, let's talk about DNA replication in general. We're gonna watch a little video, hopefully. Like I said, I think the internet is really slow, or Clark College is really slow, or my computer is really slow. I'm not sure which. Up here it comes. This might be a commercial to begin with. Nope.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right. Any questions about this this movie, video, whatever? I think I'll leave this open because we have another video to watch. Go back here. That's right. It jumped its position. Come on, mouse. Oh, there it is. For some reason, my mouse wasn't working. All right, so let's talk about DNA replication in more detail. DNA is copied by the enzyme DNA polymerase. New DNA is only made in the direction 5' to 3'. The DNA replication is initiated by an RNA primer, and the leading strand is synthesized continuously because it runs in the 5' to 3' direction. Let me see where I've got that. Right here. So this strand, if things are working well and everything is lucky, once the DNA polymerase binds to this template over here, if it never falls off, it can actually make the leading strand, the new strand, continuously without ever falling off. So if this DNA polymerase were to begin at the edge of the chromosome over here, it could theoretically, in one swoop, copy the entire chromosome. That is theoretically possible, but usually the DNA polymerase will fall off, and so the new strand of DNA would only be to something like here, from this being the edge to here. And what happens is another DNA polymerase will bind, and it'll bind around here, and then continue synthesizing this template strand of DNA. Now, you don't need to know much of that. I'm just saying that the leading strand may have more than one stretch of DNA, meaning it may have more than one DNA polymerase making the new strand of DNA to the leading strand. The leading strand is the template, by the way. And the new strand will be the newly replicated strand. The point is that the newly replicated strand may be only one piece of DNA, but then again, it could be more than one if more than one DNA polymerase binds to the leading strand. And there usually wouldn't be more than one unless this DNA polymerase falls off. And DNA polymerase does fall off from time to time. And it's not a problem because another DNA polymerase rebinds and then just continues the process. Any question about any of that? Anyways, in the leading strand, the synthesis of DNA is very easy because the strand is in the right orientation for the DNA polymerase to copy the strand and make the new DNA, strand of DNA. The other strand, the lagging strand, let me blow this up, is in the wrong orientation. And the DNA polymerase cannot move this way down the strand when the helicase opens up the DNA, which is the, it converts the double-stranded DNA to single-stranded DNA, and only the single-stranded DNA can be replicated. So you need the helicase, that enzyme is essential. You need the DNA polymerase, that enzyme is essential. And then there's a third enzyme I haven't talked about yet that you do need to know, and it's the enzyme ligase. And let me get to the end of the story, and then I'll talk about the third enzyme you must know. The RNA primer, we're going to talk about it, but it's not critical that you know this because the RNA primer puts in a stretch of RNA to begin the DNA replication. So let's say this DNA polymerase, come on, mouse, were to just begin right here, meaning this strand has already been synthesized. So there would be an RNA primer down here, and then an RNA primer will start the DNA replication. So it'll put in the RNA and then put in the DNA. That RNA primer has to be removed, and another DNA polymerase puts in the correct nucleotides for the stretch of DNA right there. And like I said, we're going to talk about it, but I'm not going to really quiz you on that part because it's not going to be a permanent part of the DNA. And I'm not sure why, but DNA replication is always started by putting in a small stretch of RNA, just a few nucleotides long, that is called the RNA primer. And then that RNA primer will be removed and filled in with DNA. Any questions about any of that? All right, on the lagging strand, since the DNA polymerase cannot move down this way, because this way is not 5 to 3 prime, the DNA polymerase has to always move in the 5 to 3 prime direction. So what it does is, let's start from this one here, is it will bind to the template and then move backwards in a short stretch on the template, synthesizing the new strand of DNA in short batches. And these short batches are called Okazaki fragments after the doctor who discovered them. And so the lagging strand is made in short batches backwards, meaning we'll have a DNA polymerase make this strand here, another one make this strand here, another one make this strand here, another one make this strand here, one binding here, making one there. So the lagging strand is made by many DNA polymerases and will be made in many stretches, small stretches of DNA. Each stretch of DNA will not be linked together to the previous stretch of DNA with a chemical covalent bond, meaning the DNA polymerase, let's talk about this one here, will begin, and then it'll come up to this one there. So we'll fill in that base pair right there, but it will not link this base to this stretch of DNA with a chemical covalent bond. The DNA polymerase does not do that. If this DNA is already made, the DNA polymerase does not link it to the base in front of it. It only makes the chemical covalent bases when the DNA polymerase puts in the first DNA, come on mouse, right here, and then it puts in the second DNA polymerase. It does link that with the chemical covalent bond, and that's because the phosphate that it's putting in, the diphosphate, gives it the energy it needs to link those bases together. Come on, mouse. My mouse died. All right. So when it puts in this one here, it uses the diphosphate to link this one here to the previous base. And then it has nothing to link this base here with the existing Okazaki fragment. So there will be a gap right there and a gap right there. So in reality, each Okazaki fragment will have a gap in the lagging strand of DNA, in the newly replicated lagging strand. At each Okazaki fragment, at the beginning of the Okazaki fragment, come on, mouse, and at the end of the Okazaki fragment. What fills in that gap, meaning makes the chemical covalent bond between this nucleotide of DNA to the previous Okazaki fragment is the enzyme ligase. The DNA ligase works a lot on the leading strand because it has to link together all the Okazaki fragments. Any question about any of that? The DNA ligase may not have to work at all on the leading strand. If the DNA polymerase begins on the edge of the chromosome over here, and the DNA polymerase moves down the leading strand and never falls off to the end of the chromosome, then the newly replicated strand will be made continuously and there will not be a gap. And the ligase doesn't have to work at all. However, as I mentioned, the DNA polymerase usually falls off a couple of times, not, a dozen times, but just a couple of times on a chromosome. We're talking about human chromosomes or a bacterial chromosome. In which case, when the new DNA polymerase binds, let's say this one falls off, this one will bind here, and then it'll start there. It won't link the previous stretch of DNA to the newly replicated stretch of DNA. So the DNA ligase will have to bind to the leading strand and connect this strand of DNA to the strand of DNA that'll be built here. But the point is the DNA ligase only has to work a few times or maybe never, on the leading strand, and has to work a ton of times on the lagging strand. Anyways, that's what DNA ligase does, and that's the third important enzyme. So you should know DNA helicase puts in the new base from the template template strand, and then that adds it to the growing strand of DNA on the newly replicated strand of DNA. Then there's the enzyme helicase. I should have started with that one. It's critical because it separates the two strands of DNA, making them a single strand, so the DNA polymerase can then bind to the single strand and use it as a template to make the new strand of DNA. And then the third enzyme that's critical is the helicase, which binds the previous strand of DNA that's replicated to the newly replicated strand of DNA, which on the Okazaki fragment, it's doing it twice for each Okazaki fragment. At the start of the Okazaki fragment, let's show it here, the start of the Okazaki fragment, and then the end of the Okazaki fragment. On the leading strand, only where there's a gap in the DNA does the ligase have to work. All right, any questions about DNA replication? So DNA is only made in the direction 5 to 3 prime. It's initiated with an RNA primer. One strand is synthesized continuously because it's in the direction 5 to 3 prime. The other strand, the lagging strand, is synthesized discontinuously and is made backwards in 5 to 3 prime direction, resulting in many Okazaki fragments. The Okazaki fragments, as well as any gaps in the leading strand, are then joined together by DNA ligase. Any question about any of that? All right. If not, let's move to RNA transcription. Transcription is the synthesis of RNA from DNA. DNA is transcribed to make messenger RNA, tRNA, or rRNA. When the RNA polymerase is transcribing the RNA, or transcribing the DNA to make the RNA, how does the RNA polymerase know to make messenger RNA, tRNA, or rRNA? Does anyone know? Nobody has a guess? It's actually very simple, and that is the RNA polymerase doesn't have to do anything. It just copies the DNA, and the gene itself will determine whether messenger RNA, tRNA, or rRNA is made. Meaning if the gene codes for a protein end product, the RNA polymerase will copy that gene as messenger RNA. If, on the other hand, the gene codes for tRNA, the polymerase will copy the gene and it'll be tRNA that it makes. And on the other hand, if the gene encodes for an rRNA product, the RNA polymerase will translate the gene or transcribe the gene as rRNA. So it really just depends on which gene the RNA polymerase is transcribing. The only difference between messenger RNA, tRNA, and rRNA is the sequence of nucleotides. You have a handful of nucleotides, or a handful of sequences which determine tRNA, RNA, another handful which determine rRNA sequences. And then we have many different sequences that determine that it's a messenger RNA. And that's because you'll have a sequence of messenger RNA that'll differ for each protein that the genes encode. So for each different protein, we'll have a different sequence of messenger RNA. Any questions about any of that? All right. Let me state that, excuse me. Ah. Some reason I had a urge to sneeze there. I could feel it coming. That's why I said, excuse me. Hopefully I won't sneeze again. Usually I do sneeze 2 times in a row. Oh, this is a little different, and that is not all of the DNA is transcribed. When we're looking at DNA replication, all of the DNA will be replicated. But when we're looking at transcription, not all of the DNA will be transcribed. Only the genes will be transcribed. Now, for a bacteria, most of the DNA does encode a gene product. So most of the DNA will be genes. There will be very little DNA, which will be non-coding DNA. However, in a eukaryote cell, most of the DNA is non-coding DNA, meaning most of the eukaryotes will not be genetic information. They will not be genes. It'll be the DNA in between the genes. Any question about any of that? All right. Transcription begins when the RNA polymerase binds to the promoter. The promoter is a region of DNA upstream of the gene, and the function of the promoter is it codes for the RNA polymerase to bind here. So that's why the RNA polymerase binds at the promoter. Transcription then proceeds in the 5' to 3' direction. So the RNA polymerase will move down the DNA in the 5' to 3' direction. And RNA will only be made in the 5' to 3' direction. The RNA polymerase will read the DNA, transcribing it. And like I said, it will only do this in a gene. And that's because the promoter is upstream of the gene. And then the RNA polymerase will transcribe down the DNA, transcribing the gene in the 5' to 3' direction. But when the RNA polymerase reaches the terminator is a region of DNA that codes for the RNA polymerase to fall off. And then the RNA polymerase falls off the DNA, and then that'll stop transcription. So upstream of a gene will be a promoter, a region of DNA called the promoter, encoding for the RNA polymerase to bind to it. And then downstream of the gene, usually right after the gene, is the terminator, a region of DNA that codes for the RNA polymerase to fall off. And that's why only the genes are transcribed. Any question about any of that? Here we're looking at an actual picture of transcription. There's the RNA polymerase. There's a little stretch of RNA being made. That's a stretch of DNA there. And this is a not really an electron microscopic image. It's a, what is it? Something fluorescent image. Any question about any of that? It is similar to an electron microscope because you can see the DNA here, and you can't see the DNA you can't see it like this in a light microscope. Any questions about any of that? All right, just a minute here. I'm gonna blow my nose. Boy, that's a bad sign. I first sneezed and now I have to blow my nose. Hope I'm not coming down with something. All right, this is showing you transcription all in one step. Here's the DNA. The RNA polymerase binds to the DNA and puts in the corresponding RNA nucleotide that actually binds to the DNA. We'll talk about that. I'll talk about this in a little bit greater detail in individual slides, but let's watch a little video. I don't think this link works anymore. Yeah. No, that's the wrong video. Okay. Get that to stop. This is our next video. Yeah, I got
The
it.
E.
Stop.
coli
Nope,
lac
didn't get
operon.
it. Stop. There we go. Don't ask me why it was playing over there. All right, let me look. I've tried that one. It wasn't a very good one. So let me look for a better one. Maybe the start of this one.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system.
Skip this.
Continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding set.
We'll talk about that later. The exons and the introns. All right, any question about any of that? Okay, we'll watch a little bit more of this video later. I think it's going to continue with translation, so we can keep it for that too, although I think my translation video is actually working. There we go. Let's go back to the PowerPoint. Come on. Why do I have two things here? I think this is changing the way this has worked, this PowerPoint. It used to, when you played the slide, it didn't keep an old copy. And so I now have to go back. This is the slide we're on. Or maybe that one. All right, let's talk about transcription. It begins when the RNA polymerase binds to the promoter of DNA. And before that can happen, I guess I should mention that the RNA polymerase can only bind to single-stranded DNA. So you need to get in a helicase here and, open up the DNA and make it single-stranded. In eukaryotes and bacteria, usually only one strand of DNA will be transcribed. But in viruses, which happen to be double-stranded, both strands could code for a gene product. But usually in bacteria and in eukaryotes, only one strand will code for the gene product. And so only one strand will be read by the RNA polymerase and then transcribed into messenger RNA. You can sort of see it here. Let me blow that up. And I didn't notice it in the little video. And that is initially when the RNA polymerase binds to the DNA where it's single-stranded, the RNA is inserted, and then the RNA is actually pairing to the DNA. And there we can see, I don't know what color that is, maybe turquoise. That's the growing RNA. And there's the RNA polymerase binding to the DNA, which in this portion right here is single-stranded. And that's because the helicase is right here, opening up the DNA and making this portion of the DNA single-stranded. Initially, when the RNA polymerase puts in the RNA, it is putting in the corresponding nucleotide, RNA nucleotide, to the DNA nucleotide. And so that RNA nucleotide will actually hydrogen bond to the DNA. And initially that does happen. So for in this portion of the RNA molecule, the RNA is actually hydrogen bonding to the DNA. Any questions about that? But someplace down the line, the RNA falls off the DNA, someplace around here, and is no longer hydrogen binding to the DNA. That is kind of important because the DNA, as the helicase moves down, making this region single-stranded, this portion of the DNA starts snapping together. And so let's say the RNA polymerase began here. This would be single-stranded initially. Actually, it had to begin right here because there's the stretch of RNA. The DNA snaps together right here. And this just is hydrogen bonding, so you don't need anything for the DNA come back together and then hydrogen bond, because the nucleotides are the correct nucleotides to hydrogen bond with each other. Anyways, any question about that? So initially, the RNA, when it's inserted, it actually is hydrogen bonding to the DNA. And that occurs for a short stretch before the RNA, for whatever reason, stops hydrogen bonding, then falls off. The Messenger RNA is then transcribed as the RNA polymerase moves down the gene. And as you can see, this whole region here, the DNA is snapped together. Only this portion here, the DNA is not snapped together. And then in this portion here, the RNA is hydrogen bonding to the template DNA. This will continue until the RNA polymerase reaches the terminator. The terminator is at the end of the gene. The terminator codes for the RNA polymerase to fall off. When the RNA, when the RNA polymerase falls off the DNA, that will release the RNA from the DNA. And then the DNA will snap back together. That is how transcription works. Any question about any of that? All right, if not, that's it for transcription. Let's move on to translation. And while we're here, let's go ahead and watch the rest of that little movie that I had, if I can find it. This is going to start talking about complication. I will talk about the complication after I discuss translation.
Actions called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different...
We'll discuss that later, not in this lesson. Okay, any question about protein translation in this video? This video did go into a few complicated factors, and I will talk about the complication for eukaryotes and how the introns are removed from the messenger RNA, but we're going to talk about protein translation first. Any questions about this video? All right, translation is the synthesis of proteins by the ribosome using the information in the messenger RNA. Translation does require the messenger RNA, ribosomes, and then tRNA. Messenger RNA is translated in groups of 3 nucleotides that we call codons. And I've got them shown in the next slide. Translation begins when a messenger RNA, or I should say the ribosome, moves down the messenger RNA and encounters the start codon. The start codon is the codon AUG, meaning the 3 nucleotides A, U, and G in RNA. The start codon also encodes the amino acid methionine. So the start codon does 2 things. It starts the process of protein translation, telling the ribosome begin protein translation here, and then it adds in the amino acid methionine. Translation will continue down the messenger RNA until the ribosome reaches one of the stop codons. And you should know the start codon because there's only one, AUG. However, you don't need to memorize the stop codons because there's 3 of them. And once the stop codon is reached, protein translation stops. Any question about any of that? All right. So when the messenger RNA is coding for the amino acids, it does it by 3 groups of nucleotides that we call a codon. And then each amino acid has, 3 nucleotides that encode the amino acid. Each messenger RNA codon codes for 1 amino acid, but organisms, at least humans and most bacteria, only contain 20 different amino acids. But there are 64 possible codons. Why? What do you do with the extra codons? Well, what is done with the extra codons is that many of the amino acids have more than 1 codon coding for each amino acid. Let me blow this up. Let's try it down here. Nah, that's not going to work. I guess I got to do it here. There are only 2 amino acids where there's only 1 codon coding for that amino acid, and that is AUG, codes for only the amino acid. Come on, mouse, start working. Only codes for the amino acid methionine. And as I mentioned, AUG also codes for start protein translation here. The other is tryptophan. There's only 1. My mouse isn't working again. There's only 1 codon that works for codes for that amino acid. And I can't show you right, it is on my mouse because my mouse doesn't work. There it is. UGG, you don't need to know that, is the only codon that codes for tryptophan. All of the other amino acids have more than one codon code for that same amino acid. For example, phenylalanine has two codons coding for this amino acid. And then serine, amino acid serine, has four codons coding for that one amino acid. And then some amino acids, like leucine, has 6 codons coding for that one amino acid. So there can be more than one codon for each amino acid. In fact, except for 2 of the amino acids, there is more than one codon coding for those amino acids. This is known as the degeneracy of the code. Sorry. Trying to end this. There we go. We've already shown the translation video, so I'm not going to show you another translation video. But let's go through the steps of translation. Translation is the translating the information in messenger RNA into protein. If you remember, the DNA is transcribed into either messenger RNA, tRNA, or rRNA. The rRNA and the tRNA is not translated into protein. The rRNA and the tRNA are used along with the messenger RNA to translate the messenger RNA into protein. And how that happens is the ribosomes will come together and bind to the messenger RNA, usually upstream of the AUG, so someplace up here. And then the ribosome moves down the messenger RNA until it reaches AUG. And then AUG starts protein translation at this site here. So the messenger RNA upstream of AUG will not be translated into protein. And that might be just a few nucleotides as here, or it could be many nucleotides, which is usually the case. Anyways, the rRNA is in the ribosome. If you remember, ribosomes are composed of rRNA and ribosomal protein. The two together make up the ribosome. The tRNA is what brings in the correct amino acid. So the anticodon UAC binds to the codon AUG, and the tRNA is what really is reading the messenger RNA. The ribosome is just moving down the RNA and then providing a spot for the tRNA to come in and then bind to the messenger RNA. Anyways, the tRNA brings in the correct amino acid, which in this case, for AUG, the correct amino acid will be the amino acid methionine. Any question about any of that? And that explains messenger RNA, ribosomal RNA, and tRNA are all important in protein translation. Let me shrink this a little. All right, this picture is only showing you 3 of the sites in the ribosome. There is a third site right here, which the video talked about, and we're not going to talk about the third site right here. That's the site where the tRNA falls off the messenger RNA. But there are 2 sites in the ribosome, and as you recall, there are actually 3 sites in the ribosome. And the first site will be where the first tRNA will come and bind to the codon, which will always be the start codon, because that'll be the first that's where the messenger RNA will start protein translation. And the second site of the ribosome allows the second tRNA to come in, and it will be the tRNA that has the anticodon right here, which binds to the codon right there. And this will differ depending on what codon this is. Now let me shrink this down a little bit here. All right, so the protein translation now has 2 tRNAs in here with the 2 correct amino acids. The next thing that happens is the ribosome takes the amino acid off of the tRNA in the first site here, which in this case is methionine, and then binds this amino acid with a chemical covalent bond to the amino acid on the second tRNA here. And this isn't discussed in your book, but this is the one place where we'll actually cover where an enzyme is doing this. And this enzyme that's doing this is actually a ribosomal enzyme. So it's part of the ribosome and part of the rRNA, part of the ribosome that removes the amino acid from this tRNA and then binds it to the amino acid in this second position here, creating the start of the growing polypeptide chain, which will be the growing protein, because when it's all done, it'll be the protein. Any question about any of that? All right. When that happens, this ribosome actually moves down a site, and then what was in this first site becomes the exit site, which we're not going to cover. But the tRNA, the first tRNA, can then fall off because it doesn't have an amino acid here. This tRNA then has the ribosome move down. Let me show that here. And so what used to be this site is now that site because the ribosome has moved down 1 codon. That will allow the third tRNA to come in. It comes in this site here and then binds the anticodon, binds there to the codon, bringing in the correct amino acid. And then once again, the enzyme in the ribosome removes the amino acid that's bound to this tRNA and then binds the amino acid to this newly added amino acid. That will allow the tRNA to fall out of the ribosome, which will allow the ribosome to move down 1 position, 1 codon, which will then allow the next tRNA to come in and bind to the codon down here. And this process will continue as the ribosome moves down 1 codon at a time, taking off the amino acid here and then binding it to the newly added amino acid. And you'll note that AUG later in the messenger RNA codes for the amino acid methionine. In this case, it's not coding for the start of protein translation. Any question about any of that? All right, so this process will continue with the ribosome moving down the codon. allowing the tRNA to come in and then read the codon on the messenger RNA, bringing in the correct amino acid. And as I stated, we say that the ribosome reads the messenger RNA. The ribosome doesn't. It's actually the tRNA which reads the messenger RNA. But the ribosome allows for protein translation to happen, and this is where protein translation occurs, at the ribosome. And you do need the messenger RNA and the tRNA to come in and help in protein translation. This continues until the ribosome moves down to a stop codon. The stop codon tells the ribosome to stop protein translation here. And so protein translation stops. And what happens is the ribosome will release the amino acid from the tRNA. That tRNA will fall out, and then the ribosome will dissociate and fall off of the messenger RNA, as shown here. When that happens, the tRNA, which previously was only in the primary structure, all it was a sequence of nucleotides, sequence of amino acids. The sequence of amino acids will then start interacting with each other, and you'll very quickly get the second level of a protein structure happening. Anyways, when that happens, this will be the newly translated protein, which we call the protein. Before that, when it's the growing protein, you really should call it a polypeptide chain. All right, any questions about protein translation? Any questions? Because I'm going to get into a complication now. The first complication is that in both prokaryotes and in eukaryotes, you can have more than one ribosome bind to the messenger RNA. So right here we have one messenger RNA, and there's 1, 2, 3, 4, 5 different ribosomes bound to it, making 5 different proteins, or growing polypeptide chains, I should say, which isn't shown here. It's not even shown here. This is the actual electron microscopic image. And this complication happens in both prokaryotes and in eukaryotes. You can have more than one ribosome bind to the same messenger RNA. In prokaryotes, we have an additional complication, which is shown here and here. And what's happening is, as transcription is making the RNA, protein translation is already beginning. Like right here, that clearly is not a finished RNA transcript. The messenger RNA is shorter here than here, and so the messenger RNA is still being made, meaning that protein translation is happening at the same time that RNA transcription is happening. And this complication is shown here, and that can happen in prokaryotes because both transcription and translation happen in the cytoplasm of prokaryotes. That doesn't happen in eukaryotes because in eukaryotes, transcription happens in the nucleus and then translation happens in the cytoplasm. Any question about any of that? All right, let me talk about the last complication, which is seen in eukaryotes. And that is, in eukaryotes, we have in DNA regions of the DNA that code for the protein, and we call those regions of the DNA an exon. But we also have regions of DNA that do not code for the protein, and that we call introns. when that messenger RNA is initially transcribed, it'll have both exons and intron regions. This initial RNA will not be allowed to leave the nucleus. The pores of the nuclear membrane will not allow the messenger RNA to go through if the messenger RNA has introns in it. The introns have to be removed. And how that happens is they will be spliced out. That means cut. The introns will be cut out, and then the exons will be spliced together to make the final finished messenger RNA that lock the introns. And this final messenger RNA lacking the introns will be allowed out of the pores in the nuclear membrane. And then they'll go out in the cytoplasm to begin protein translation. Anyways, that's the complication of protein translation in eukaryotes. Any question about either of these complications? Any questions about protein translation? All right. If not, I'm going to stop here and I'll see you at 6:30 for the lab. Bye.